Dear students, today we are going to discuss about the podocytes. Why we are going to discuss about this cell? Podocytes are basically the important cells which are responsible for some kind of clinical significance diseases. Now, what are the diseases? These diseases are number one, minimal change disease, and the second disease is the focal segmental glomerulosclerosis. Podocyte is a very important cell which is responsible for the filtration and which is responsible for the formation of the urine. It is the very important cells which help in the formation of the urine with some other structures of the Bowman capsule or glomerulus. Basically, podocytes are highly differentiated epithelial cells. Podocytes are basically not replaced by the other cells. So many progenitor cells have been tried to develop the podocytes, but all trials are unsuccessful. Now, we are going to discuss about the podocytes, what the podocytes do and how are basically belong to a particular kind of structure. When we see a podocyte is basically the visceral epithelial cells of the Bowman capsule. As we know about the Bowman capsule, we are not going to discuss about the Bowman capsule here. We are not going to discuss about the glomerulus, as we know about the glomerulus and we know about the Bowman capsule also. We are basically talking about the podocytes only. And as we know, the podocytes are the visceral epithelial cells. Visceral epithelial cells, which are highly differentiated, which are called the podocytes, have the two main structures. One structure is called the cell body and the other structure is called the food processes. And this basically cell body emerges as a primary processes which ultimately ends into a food processes. Now, how this basically works and how is this structurally placed? Basically, podocyte has a cell body. What is cell body? For example, this is the cell body, this is the cell, this is the podocyte, and this is the cell body. Podocyte's cell body is like other cells. This cell has Golgi apparatus, endoplasmic reticulum, microfilaments, other filaments, microtubules, etc. So like other cells, this is the cell body, and with this cell body comes out a primary process, and primary process, this ultimately leads to the food process. So this podocytes has a cell body and a food process. These food processes are very important. And how they are important? For example, this is the capillary. This is the capillary of the glomerulus, and this capillary has a basement membrane. And this basement membrane has basically kind of collagen fibers, which we call alpha 3, alpha 4, alpha 5, etc. And so these podocytes food process attach to this basement membrane like this. This is the food processes and they attach to the basement membrane like this. But by the side, these food processes make the interdigitation with the other podocyte, and they form the structure like this. So at one part they attach like this. This is the food process which is attached to the basement membrane, and by the side they attach with the other podocyte, which are called the interdigitations, and these interdigitations basically form the slit diaphragm, which we call the slit diaphragm. And this slit diaphragm is very, very important. Now, cell body which forms the primary process from the food process attached to the basement membrane, and these food processes also make the interdigitation with the other podocytes, and which we call the ciliate diaphragm. When we see these ciliate diaphragms under the conventional electron microscope, we are not able to see these ciliate diaphragm. But when we use the transmission electron microscopy and stain this podocyte with the glutaraldehyde, we see a bridge, like a dark line. So when we use the glutaraldehyde, we see a dark line in a transmission electron microscopy. But when we stain with glutaraldehyde plus tannic acid, we see these ciliate diaphragm like a zip. So these interdigitations are like a zip. So by the side, this is the basement membrane. By the side, there is a ciliate diaphragm, and one side, these podocytes are anchoring with the basement membrane. Now, this food process. Now we come to the point how these food process of the podocyte anchor with the basement membrane. How these podocytes food process anchor with the basement membrane. When we see the structure of the food processes have some kind of bundles, and these bundles are basically made up of a kind of protein which we call beta-actinin protein. These beta-actinin protein are usually made these bundles, and these bundles are in the form of U shape. So these podocyte food processes are made up of some kind of bundles which we call beta-actinin bundles, and these bundles are in the form of U shape. These bundles are overlapped with some other filaments or microfilaments, and when they come into the dense cytoplasm outside of the food process, they anchor with some kind of proteins, or you can say some kinds of dense particles. Or what are these particles and what are these, you can say, dimers. Basically these are called the dimers, and there are two dimers which connect this beta-actin protein to the basement membrane. These dimers are, number one, alpha-3 beta-1 integrin dimer. One dimer is called alpha-3 beta-1 integrin dimer. This alpha-3 beta-1 integrin dimer connects with this beta-actinin protein by three proteins which we call vinculin, paxillin and tallin. Vinculin, paxillin and tallin. And they connect with the alpha-3, alpha-4 and alpha-5 chain of the basement membrane respectively. Clear? So now a connection is made. So the U-shaped bundle of the beta-actinin followed by with the dimers, alpha-3 beta-1 dimers. Alpha-3 beta-1 dimers uses three proteins, vinculin, paxillin and tallin, and they connect the corresponding the collagen chains alpha-3, alpha-4, alpha-5 of the basement membrane, and they anchor with the basement membrane. They anchor like this, floating. So, and likewise some other dimers like alpha-beta dystroglycans, they are also dimers, and they use some other proteins like eutrophin and agrin kind of proteins which they use, and they make this anchoring. So this is the process, how they anchor with the basement membrane. Now, they now anchored with the basement membrane. Now we talk about the food process which make interdigitations with the side, by the side of the podocyte, other podocytes. And some proteins are responsible for formation of these interdigitation or zipper-like structure, or which we call the slit diaphragm, which we can see only in the transmission electron microscopy stained with the glutaraldehyde plus tannic acid. So, what are the proteins? These proteins are, number one, nephrin, podocin, CD2AP, FAT1, and some other proteins also. So nephrin, podocin, CD2AP, and FAT1. So these are the proteins which are basically responsible for the formation of this slit diaphragm. Why these proteins are important? Why this anchoring is important? Because these have some clinical significance. If there is disturbance in the protein like nephrin, there is a disease which we call congenital Finnish type nephrotic syndrome. Congenital Finnish type nephrotic syndrome. Other disease which is related to the podocin, which we call the minimal change disease and focal segmental glomerulosclerosis. So these proteins have clinical significance. Whenever these proteins are absent because of some genetic abnormalities, so we see these absence in the form of these kind of congenital diseases. Now, after formation this slit diaphragm, we discuss about the receptors on the podocytes. Look, podocytes are the cells like other cells, so they have receptors. As the other cells have receptors, the podocytes also have receptors. The most important, like other cells, is the cyclic GMP. Cyclic GMP receptors are basically stimulated by atrial natriuretic peptides, brain natriuretic peptides, and C-type natriuretic peptides. Likewise, some other receptors are like cyclic AMP, and cyclic AMP are stimulated by prostaglandins, prostaglandin E2, VGEF, and parathyroid hormone or parathyroid hormone related proteins are responsible for the stimulation of these receptors. And how they stimulate, how they function, how they what they do, we are not going to discuss here. But these are the receptors which are found on the podocytes. Another are calcium sensing receptors, and the calcium sensing receptors are stimulated like acetylcholine. They are stimulated by acetylcholine. They are stimulated by angiotensin 2. They are stimulated by ATPs. They are stimulated by histamine. They are stimulated by endothelin, etc. So these are the receptors which are being stimulated. Now, beside these receptors, these podocytes also have some kind of channels, which these channels are now getting some kind of importance in function of the podocytes. And what are the channels? The channels are like TRPC5 and TRPC6. So in understanding the congenital disease of this kidney and the congenital disease of the some form of nephrotic syndrome, we are basically, scientists are basically trying to understand the role of these TRPC5 and TRPC6. So students, this is the basic concept about the podocytes. Podocytes are very complex structures. Podocytes in some kind of diseases, when we do the renal biopsy and we see the structure under light microscopy or immunofluorescence, we do not find any abnormality. And sometime in electron microscope also we do not find any abnormality. And some kind of, you can say, effacement is found there. And this effacement, why is there this effacement? We usually not able to understand other than some theories. So some factors which are now known and which are unknown are responsible, and which we call the permeability factors. These permeability factors are responsible for the disturbance of these structures of the podocytes, especially the slit diaphragm, and responsible for the diseases like minimal change disease and FSGS. These diseases are very common, and these diseases are basically responsible for the loss of the proteins in the urine. When this slit diaphragm, which has the diameter of around 30 to 40 nanometer, is being disturbed by any cause, whether the primary cause or some secondary causes. What are the primary causes and what are the secondary causes, we'll discuss later, of these diseases. So when this slit diaphragm is disturbed by some primary causes or some secondary causes, we lose the protein because we lose the diameter. What is the diameter? Diameter is around 30 to 40 nanometer. So when we lose this slit diaphragm, the diameter of the slit diaphragm, the proteins come in the urine. And when the protein come in the urine, we found the deficiency of the protein in the blood. And we found that there is a kind of swelling, kind of features of nephrotic syndrome. And this nephrotic syndrome is very common in children, no doubt, as well also in adults. But nephrotic syndrome is very common when these proteins come out after the disturbation of this slit diaphragm and other structures of the podocytes. So, in lecture series of the podocytopathy, we are going to discuss the disease, minimal change disease, in next lecture, and we'll discuss how the minimal change disease is being categorized, what are the etiological factors, pathogenesis, treatment, etc. So this is the basic concept about the podocyte, and if we understand the podocyte, we can understand basically the related clinical significance of the diseases like podocytopathy. Thank you.